Okay, we're going to start a new unit on genetics, and this will be basic genetics. And we're looking at chapter 11, section 1, and we're looking at the work of Gregor Mendel. So think about it. What exactly is inheritance? Well, inheritance is something that we receive from our parents, and it could be anything from your hair color to your skin color to whether or not you have freckles, whether you have little points on the ends of your ears, things like any of the traits that you would receive from your parents. As far as the definition of genetics, genetics is considered the study of heredity. There was a scientist named Gregor Mendel, and he's known as the father of genetics. And he was born in 1882, and he was a priest from the Czech Republic. And in college, he studied science and math. And he was in charge of the garden in the monastery where he lived, and that's where he was able to do his experiments that would change biology forever. So look at the year, we're talking 1882. So this is a little bit after the microscope was invented. So in his experiments, he used garden peas, and garden peas are considered what's called a model system. And they're considered a model system because they grow fast, they're easy to grow, they're small, and you can get hundreds of offspring. Fruit flies are also considered a model system. So when he did his experiments, first thing he looked at was what role fertilization had in it. And in order to do this, in order to fertilize the plants, he had to know the male and female parts of a plant. So pretty much the male parts produce the pollen, and the female parts produce the eggs. Okay, and the name of the male parts are stamen, and the name of the female part is the pistil. So a couple of terms that you're going to need to be familiar with. First one is called true breeding, meaning that when you breed these plants with the same type of plant, you'll get identical ones. So they produce offspring that are identical to the parent. So if you have pink flowers and you fertilize them with other flowers, you'll keep getting pink flowers. And that's considered true breeding. Trait is another term that you'll have to know. And basically what a trait is a characteristic. So a trait could be anything from hair color to skin color to eye color to... Okay, so when we're looking at the peas, or when Mendel was looking at the peas, he noticed that they're normally self-pollinating. In other words, the male parts will pollinate the female part on the same plant. So he decided to use what's called cross-pollination. And cross-pollination is when you take a brush, or you can even take your finger sometimes, and you touch the male part which has the pollen on it, and you get some of the pollen off, and then you go to a different plant and touch the female part with the pollen. You place the pollen from the one plant onto the female part of another plant. So when Mendel was doing this cross-pollination, he was looking at 7 different traits, and here they're on this table here: seed shape, seed color, seed coat, the pod shape, the pod color, the flower position, and the plant height. So with the seed shape, you'd either have round or wrinkled ones. Seed color, yellow or green. When we're talking about the seeds, we're talking about the actual peas. The seed coat, in other words, the color of the outer covering would be the gray or white. The pod shape, it'd either be one smooth cylindrical shape or be constricted. The pod color could be either green or yellow. And then the flower position could either be axial or terminal. So when doing genetic crosses, you're looking at the original, or the first group that you do would be considered the P generation, or the parental generation. Okay, so in our case, in his case up here, it's the top row here, round and wrinkled would be the P generation. Yellow and green would be the P generation. So you had one plant that was yellow, seeds and one with green seeds. That's the parent generation. The offspring would be called the F1, or the first filial generation. So that's the second row here on this chart. Okay, so if we still look at the round and wrinkled, in the F1 generation, they all came out round. And that's assuming we started with true breeding round and true breeding wrinkled. So for each of these traits, okay, he took the true breeding version of it and he cross-pollinated them. These were the results that he received. So he came up with two conclusions. The first one was that the characteristics are determined by factors that are passed from one parent to the next. Today we call these factors genes. We know that genes get passed. And the different forms of a gene are called alleles. So you might have an allele for round and an allele for wrinkled. But the gene would be the seed shape. His second conclusion was the principle of dominance. And it states that an organism with at least one dominant allele for a particular trait will exhibit that form of the trait. So if the dominant one is present, the dominant allele is present, that's what you're going to see. So here's a picture of like a pink looking leaf and a greenish one. And the dominant one obviously is a pink one because that's the one that's expressed. That's the one that's exhibited. So looking at this chart, which traits were dominant? Well, the purple flowers were axial flowers, yellow seeds, round seed shape, the smooth or the inflated pod shape, the green pod color, and then the tall stems. Okay, so the offspring of the F1 cross are called the F2. The offspring of the F1 are called the F2. Now if you look at these, you had a tall one, true breeding tall, with a true breeding short, and all of the F1 came out tall. Now remember, they've got the trait for short in there. So when you crossbreed or cross-pollinate those, you ended up with some tall ones and a short one. So this is where Mendel had to explain what happened here. And this is where he came up with the law of segregation. And what it states is that allele pairs separate during gamete formation. Gametes are sex cells. We talked about this in class before. Gametes are sex cells. Girls have eggs, boys have sperm. So when we're looking at the formation of the gametes, we're looking to see what trait is in the sperm or what trait is in the egg. So if you look at the tall ones here in the F1, they had, in their gametes, they would have a tall allele and they would have a short allele, which is the little t. It's denoted by the letter T. And the same thing with the other. And because of that, depending on how they segregate, how they separate and come back together, will determine what the offsprings will look like. So if they are the same, in other words, if the gametes are the same in the offspring, it's considered homozygous for that trait. So that's another new word, homozygous. If they are different, like the ones in the middle with the big T and the little t, they're considered heterozygous. Then the short one is little t, so that's considered homozygous. Homo means the same, hetero means different. So he came up with his law of segregation, and that when the gametes are produced, the alleles for each gene will segregate. In other words, they separate from one another, so they're independent of each other. And each gamete carries only one allele for the gene. Okay? Now, if the gamete or if the gene, or excuse me, if the allele is dominant, we denote it with a capital letter. If it is recessive, we'll denote it with a lowercase letter. So when you look at the F2 generation, there was different combinations of alleles, and you had a couple tall ones, and then you had one short one.